So I am very grateful to be invited to this really interesting workshop, and I've never been to a workshop with such a diverse audience, and this is really a lot of fun. It's been very thought-provoking. So I have no financial interest in any of the companies or drugs. What I will be talking about today is the role of fascia and collagen in cancer, so I do have an interest in that. And I wanted to start with a connection. I know you've seen this a lot today, but to think about what parts I'm going to tell you about. And Helene gave us a very nice example of how there's a relationship between the connective tissue fascia, which is composed of a lot of collagen, and how that relates to fibrosis and inflammation. And then I'm going to try to draw the link of that to cancer biology and how we get to formation, spreading, and metastasis. So I'm going to start by telling you a bit about how the normal breast structure is and the environment that breast cells are in and how that alters in cancer. So the breast, normally if you were to take a cartoon of it, looks like this. And if you were to cross-section through these areas, it's composed of these branching trees that the cross-section of them are very nicely organized epithelial cells that are very well behaved, and they form this nice little lumen in here, which is the open space. And this is a human biopsy of normal breast tissue. The darker parts are the collagen, or the epithelial cells, and the lighter pink is the collagen. And here's just a blown up example that I downloaded from the same place as Helene did, I noticed, of loose connective tissue and just showing you collagen fibers. Now, as tumor progresses, what occurs is this is a cancer biopsy, a pathologic sample. The epithelial cells are now interacting with the collagen, but in a different way. And so these cancer mammary epithelial cells are invading through this collagen environment. So what we know is that this environment is very important for cells to behave normally. If I were to take these epithelial cells out of this environment in the breast and put them in the similar environment of the salivary gland, they will become saliva cells and make saliva. So the environment around them tells them who they are, how they should behave. And similarly, those interactions are completely disrupted in cancer, as we will see. Now, this is a different view of this. I love this image because it really gets you thinking about the fact that this is three-dimensional. So this is a scanning electron micrograph that one of my former postdocs took. Each one of these big balls is a single mammary epithelial cell in one of these structures. And then these ropes and fibers are the collagen surrounding this structure. And when you look at this, you start to get the sense that this is not just a thing sitting there. It's got a very physical interaction with the cells. And the cells, as you just heard from Boris, have receptors for this collagen, but they also have these physical interactions with the collagen. And you might envision that the collagen even plays a restraining role. All right, so our story links to a very common finding, which is that dense breast tissue is linked to a huge increased risk of developing breast carcinoma. In fact, other than being a woman, this is the single largest risk factor for developing breast cancer. And here's an example of low and high mammographic density. And this is work that was pioneered by Norman Boyd's lab over the past probably 15 years. And in Norman Boyd's lab, they discovered that high mammographic density is largely correlated to an increase in the amount of collagen that's in that connective tissue microenvironment. So why should this be a risk factor for developing breast cancer? And that's what we've been setting out to look at for part of this. All right, so one of the ways we can look at this, we need tools, we need model systems to look at this, is what are called three-dimensional culture systems. In vitro means in glass, so anytime we're not in an animal or a person, we're in vitro. And we can mimic the branching ductal structures by putting breast epithelial cells into these three-dimensional gels. Now think Jell-O. Literally, this is Jell-O. What Jell-O is denatured collagen. There's no strawberry flavoring, but otherwise, this is native collagen, and the breast cells are forming these branching ductal trees. And you can put them in different extracellular matrix environments, and they will form more of the what we call acinar structure. That's the end part of the of the mammary gland. So we have ways of looking at this. Now, one of the things we can do is we can put our cells in these three-dimensional collagen gels and culture them for a while. And I'm showing you two images here. And these images are both from a human breast cancer cell line that's been in culture for many years. And these cells were put into a single tube of collagen that was neutralized, and it was put into side-by-side dishes. It was given all the same media and all the same serum and everything, put in the same incubator, and 10 days later, these are profoundly different. There's one big difference. The one on the left was detached from the sides and the bottom of the dish so that this gel is floating. The one on the right was left attached to the sides and the bottom of the dish. So what we have here is this one over here is contractible and compliant. The cells can literally pull on this collagen and change it. This one over here, when the cells pull on the collagen, it's stiff and restrained. And what you see, each one of these little circles is actually a cell. Here the cells are so integrated with one another, you can't tell one cell from another. And they're forming this ductal branching morphogenesis of a normal mammary gland. Here, they're invasive and they're behaving like cancer cells. Now, the interesting thing about this, these are cancer cells. All right, here's a different example. These are normal human mammary epithelial cells, and now you're looking at them a bit different, so I have to apologize. Here, what you're looking at is the cytoskeleton in green and the nucleus in blue. The collagen is around here. You can't see it. So what we're doing here is we're using this idea of stiffness that Boris introduced and Melody talked about, where as we increase the amount of collagen concentration, we increase something called the modulus. So it becomes stiffer. So here, think about the fact that if you want to make Jell-O jigglers and give them to your kids, you need to add more Jell-O so that they can pick them up and throw them around. That's what this is. So, so here we've got compliant and stiff, and in the compliant condition, these cells make very nice what we call acinar structures. They're behaving very normally and well. These guys here are behaving like cancer cells. We call this an epithelial mesenchymal transition. They are invasive, they are disorganized. Now look what we just did. We took normal cells and we made them behave like cancer just by putting them in more collagen that's stiffer. Here we took cancer cells and we made them behave like normal cells by putting them in a compliant environment. So this environment can override the genetic changes that occur in cancer. And it's very instructive. Now, how does that work? And I apologize, these are some signaling molecules. You don't need to think about them too deeply. But the idea is this. If you have a cell in a collagen gel, picture that it's reaching out, it's grabbing onto the collagen, and it's pulling. And the process it uses is a process based on the cytoskeleton called actin and myosin. And this is a muscle diagram of what's inside a muscle. And it's the same mechanism, only it's not muscle versions. And so in a muscle, you've got myosin here and actin. And the way you contract your muscles is actin and myosin slide against each other. Same thing here. Actin and myosin slide against each other. This thing, this MLC, is the thing that prevents that from happening. When it's phosphorylated by this thing, then this can occur. So basically, this Rho kinase, I'm going to call these Rho-ROCK from now on, are a signaling protein in cells that allow cells to contract like pulling on a muscle. So if you have a cell that contracts and the environment's stiff, it meets with resistance. If the environment is soft, it can pull it up like a blanket. Now we know that there's fundamentally many changes, and we can look at what we call gene expression analysis, and we can see if we have a low density or a high density, so this is soft or stiff, environment, about 3,000 genes change in these cells. And so you get cells here that behave like normal epithelial cells, cells here that behave like cancer. And here's a very complicated signaling diagram, but it basically what it says is akin to what Boris just showed you very nicely with the candy wrapper. I really love that. So these are our integrins that he mentioned. They're called integrins because they integrate the inside of the cell with the outside of the cell. They attach to collagen and other extracellular matrix proteins. On the inside, they can attach to this actin myosin cytoskeleton. And what happens is they activate this Rho-ROCK signaling pathway in this contraction. And this is a feedback loop And if you pictured that you had a bunch of balloons with strings floating around, if I were to grab all the strings and pull on them, they would come together. Now, in this case, like the candy, that will only happen if there's also force on the outside that doesn't, that resists them so that you can pull them together. Now, when that happens, a few hundred proteins assemble onto this big scaffold, and this then creates a whole bunch of biochemical changes in the cell that leads to certain factors going into the nucleus of the cell and activating these 3,000 genes. So there's this very precise way that this occurs that we're starting to understand. We don't know everything, but we can understand aspects of it. All right, so that is all putting cells in a culture system. So what happens if we go into a model system such as a mouse or going on into people? So what we made use of here is a model where the collagen gene is mutated so that it can't be cleaved and turned over. So the result is that you end up with more collagen in the mammary gland and actually in the whole animal. So this is a normal mouse mammary gland. This is fat around it, and the pink here is collagen. You can see that in white here. Here is one of these collagenase-resistant mice. We'll call them Coll mice. And what you have is you actually have more collagen around the mammary gland. And interestingly, already there's this excessive growth of the cells, even when you don't have cancer going on here. What we did is we took these animals and we crossed them to one of our favorite models of breast cancer in mice called this polyoma middle T model. So these animals all get breast cancer in the mice. But what happened when we did it in this collagen-rich background is we had faster tumors and we had more of them. But the most striking difference is when we went to take those tumors out, to excise them, they were incredibly attached inside the wall of the animal. And this is an example. So in green is the collagen and kind of in red is the tumor. In the normal mouse background, the collagen looks like this after 10 weeks of age. And in this collagenase-resistant animal, we have these really thick, dense collagen fibers. And these animals have a threefold increase in metastasis to lungs. So that means these breast cancer cells are going to the lungs and making metastases. Now, what's interesting here is this looks like a big structural change as well as an amount of collagen change. So from there, we backed up and we started thinking about, well, what's the structure of collagen? So one of the things that has been very confounding in the mammographic density field is that there are a lot of studies, and they're conflicting, about whether density predicts long-term outcome in humans. And so by long-term outcome, I mean, are you going to get breast cancer again, or are you going to die from breast cancer? So breast density is very clearly linked to getting breast cancer in the first place. But the jury is still out about what its role is long-term. It doesn't seem to predict long-term relapse. In some studies, it actually predicts good long-term outcome. So we started thinking maybe the structure is what matters and not the amount. So I'm going to back up really briefly. We're using an imaging method that is worth mentioning. It's called second harmonic generation in multiphoton microscopy. It's based on the physics of light and collagen. The only thing you need to know about this is this allows us to see collagen in any sample live without staining it. We can take pathologic samples and see collagen. We can look into live tissue. We can look into a mouse. We can look into a human biopsy. This is live rat tissue right here that has no stain. It's been taken out of the rat, plopped on the microscope. This is what we see. This is normal mammary gland, and we see all this beautiful curly collagen. So what we found in looking at a number of mouse models is that as we go from normal mammary gland through progression into early and later, and this is metastatic breast cancer, we would find these tumor-associated collagen signatures, these changes where there's a deposition of straight collagen that initially goes around the tumor stromal boundary like this, and then ultimately becomes perpendicular to the tumor stromal boundary, and we actually see cancer cells migrating out on this. We very creatively called these tumor-associated collagen signatures 1, 2, and 3. So we could talk about them. So what we did is we then went and we looked. So we saw these in mice. We then went to human tissue. These are fresh biopsies out of our tissue bank that were frozen. this is normal breast tissue. It was a benign biopsy. It's probably fibroadenoma. And you can see all this curly collagen and lots of it. This one over here is an invasive ductal carcinoma. There's actually less collagen here, but it is really thick and bundled and straight. You can tell how different it is. So what we did is we took what's called a tissue microarray. These are slides where there's little biopsy punches of 300 patient samples, and we went, this is just showing blowing them up each one. So purplish is what the pathologist sees, and then in kind of yellow is what we're seeing with our second harmonic imaging overlaid on this. And we were looking for islands of cells and the collagen at those islands. And what we did is we had 3 unsuspecting graduate students who thought they were going to do exciting stuff look at 14 regions of interest across 300 samples and go yes or no if there was one of those signatures. And then we totaled it all up and we sent it to our biostatisticians who had all the outcome data. And what we found was quite remarkable. We have a very strong ability to predict long-term outcome in human breast cancer. This goes out 200 months. That's about 20 years. And so in red is if the patient has this perpendicular collagen, any perpendicular collagen, and in black is if they don't. And so you can see it has a very good prognostic ability for having breast cancer again or for dying of breast cancer. And so this has been rather exciting, and we're taking this forward. Now, one of the things we want to do, that was invasive cancer. So that set of tissues, we already knew that they had cancer that was spreading. So that told us what the long-term outcome is. But there's a lot of situations where it's not so clear. One of them is called ductal carcinoma in situ, where patients have tumors that can be quite large, but that are completely contained within the connective tissue. Sometimes we actually do see some aligned collagen. Sometimes we see curly collagen. The thing about these patients is that 1/3 of them need no treatment at all, and the other 2/3 are likely to move on to full-blown cancer that's invasive. So this is actually a precancerous stage by many definitions. Most of these patients elect to have full mastectomy or lumpectomy and have a portion of these tumors removed. They end up getting the lymphedema we've heard about. Many of them have the very toxic chemotherapies. Now remember, 1/3 of them need nothing at all. They just need to be watched. Problem is we don't know which third. So we are looking to see, does the environment around the tumor tell us which third of the patients are going to be okay without further treatment? We are on our way through 400, 500 patients. We added to this, so we have about 600 to do. This is a work in progress. What we're finding is that there's lots and lots of different kinds of structures of collagen. And one of the things, oh, I cut it out. Okay, so one of the things we're doing is rather than asking graduate students to have to go through and call it visually, we're using machine learning approaches to try to get computers to see these different features and then to quantify things like length, curliness, width, alignment, so forth. And so we have a lot of progress in that. If you're interested, please ask me after the session. All right, so what I've just told you is that collagen contributes to metastasis. So you get a lesion in the breast, it spreads, and it does so by walking out along the collagen fibers. So how does that occur? So one of the things that happens, we can take, this is just a diagram of the experimental setup, and we can take a collagen gel, and this one's a compliant collagen gel. We can take either tissue biopsy punches or cell-seeded collagen gels and put them in this collagen gel. And the cells will actually align the collagen in between the two plugs. They pull on the collagen just like the Boris's daughter pulled on the candy to be able to open it. They stretch it out. And then what we find is that cells prefer to migrate into the regions that are aligned compared to the regions that are not aligned. And in fact, that alignment requires that they have this row rock ability to flex their muscles, as it were, to contract. So what happens here is that the cells pull on the matrix, kind of like taking a rope and straightening it out. And so if you picture that you have this rope that's curled up and you pull on it, at some point you're going to pull all the flexible parts out. You're going to end up with straight rope. And if you were sitting here on roller skates and you had a pile of rope that was attached to the wall, and you started pulling on it, at first you would go nowhere. But at some point you'd pull on that rope and the rope would be straight and taut, and then you would move towards the wall. That's our model in a kind of simplified version of what we think is happening here. Now, if you give the cells choices, so you put the cells in a gel here, and you pre-align the gel for them using, in this case, magnets, but we've used various engineering approaches, and you give them a choice. Do you want to walk along the railroad tracks or across the railroad tracks? Overwhelmingly, they prefer to go perpendicular along the railroad tracks out. They don't like to walk across these very well. And now, if you put in drugs that inhibit this Rho-ROCK pathway, you don't inhibit it at all. So the Rho and ROCK are there to allow the cell to pull on the ropes, to pull on the collagen, line it up. All right, so now what's really cool is that, so here's an example. This is the collagen here, and this is a very low resolution view of cells migrating out from the collagen. You can kind of see these are bipolar, these are more flexible. It's much easier to see this. Here we have labeled the cells with a probe for actin. It's it's the thing that interacts with myosin, and it's part of the cytoskeleton. These guys have all these different protrusions, and they're probing their environment and wandering around. And these guys are very persistently going in a linear fashion. So these guys are kind of distracted, and they're wandering around. They wander just as fast as these guys, but they get lost. They don't go to where they're going. These guys very clearly go in one direction. Now, if you put this with a blood vessel on the other side where there's growth factors coming out of the blood vessel, these guys will go straight for the blood vessel. So it's actually a rather scary phenomenon if you think about it in terms of cancer spread. So this is just quantifying this. We can look at how many protrusions there are, and if they're aligned, there's a lot fewer than if they're random, so there's fewer distractions. And we can look at how persistent they are, and the cells are more persistent in this aligned matrix. So what I'm telling you is that this aligned collagen is like a superhighway, allowing these cells to get out of the tumor and go elsewhere, and to do so very efficiently. Okay, so what happens when they get to the metastatic site? So these cells leave the primary site, they get into the vasculature, and then they have to get out at the primary site. Now, in this experimental model for our mice, our secondary site for metastasis is lungs. But in human breast cancer, it can go to liver, they can go to brain, they can go to bone marrow, they can go to lungs, and true for mice as well. This is a normal lung with no metastases. The white is the collagen, imaged by second harmonic, live tissue. And over here, we have injected into the tail vein a mouse breast cancer cell line that is labeled with a green dye, a fluorescent green dye. And so we can see it. And here's what the collagen looks like around that tumor. So when the cells get out into the lungs, what they do is they call the microenvironment into play, and they get the microenvironment to make the environment that they like that allows them to grow and proliferate. They get this fibrosis or this collagen deposition of this aligned collagen. So we are guessing, we have no real data to know this, we're guessing that some cells that go to the lungs and don't form metastases, we know there's cells that go there and don't do anything, that those cells are not good at doing this. And it's only the cells that can do this that then form metastases that are observable. All right, so to summarize this, tumors arise in dense collagen, and they are more aggressive. The collagen becomes aligned around tumors, and that alignment facilitates invasion. Okay, so what's really cool about this, in cancer, almost everything is a co-option of something that is in place for good, normal reasons. So we have the wound healing response that I'm going to show you in a moment is co-opted here. But part of the reason for this is part of a normal developmental process, we think. When the mammary gland forms, or the salivary gland, or many branching organs, the lung, It starts out as a little bud that has to invade into a connective tissue fat pad, and around that connective tissue, that invading bud, is linear collagen. And so this thing invades and branches using collagen as a template, we think, upon which it can branch. So what we think is happening is the breast cancer cells are actually going back to their embryonic state and making use of processes that they were able to do to set up the mammary gland in the first place. All right, here's a mouse picture. It's not too gruesome, so sorry about that. So one of the things we can do is we can look into the mice, and we've actually developed an imaging window that is very well tolerated and has very minimal wound response. This mouse is nude by nature. It's an immunocompromised mouse. We didn't shave it. It just is born that way. And so we can look through a little window, and we can look at tumors live. So here, white is collagen, and blue is the tumor cells that are in the window. And so here's one such image where we've got, in blue, tumor cells. You can see one starting to invade out right here. And green are all these immune cells that you've heard about today from Melody Schwartz, from Helene Langevin, immune cells that are macrophages and neutrophils. And there are a lot of them in the environment, and actually they're much more active and migratory than the cancer cells themselves. So one of the things we observe, if we look at, here we have the immune cells in green, our tumor is in red, and then the collagen is in white. What we observe is that the immune cells are coming into the tumor on the collagen fibers. And so this is not unlike what we heard earlier about stretching the bit of tissue and seeing that the neutrophils could actually migrate better when you stretch the tissue. Now, if you take these dense collagen tumors I told you about versus the wild type, we find that there are more neutrophil populations in the dense collagen tumors. We're marking these by a particular marker called Ly6G. And what we do, and when we look at all the chemical messengers that we call cytokines, we find that there's differences between these dense collagen mice and the not dense collagen mice. And what you see here are the same things, they're just inverted. So everything that's above the blue line in red is elevated in the wild type animal, or the dense animal, and everything in gray is elevated in the wild type animal. And so there's actually different chemical messengers in these two different systems. So what that suggests is that if we have women with dense breast tissue, their breast cancer might be fundamentally different than women who don't have dense breast tissue. And when we look at what cytokines we have, they're signaling, a lot of them are signaling to neutrophils and macrophages. So one of the things, and this is the work of my student Maria Garcia Mendoza, who is going to graduate next month, she depleted these cells with an antibody, and this is just showing us what we're doing to look at these is mouse PET imaging. And what you see here is these are the wild type animals, and over time, from day 1 to day 21, the tumors grow, and the dotted lines are the treatment, and the solid are the untreated. This is number of tumor and total burden. And here, if we look at the dense collagen animals, there's actually very little growth for the dense collagen animals, and there's a significant difference when we inhibit the neutrophils. If we look at the number of mice with metastases, the treated wild type animals that have the neutrophil depletion, they all have metastases. Only 1 out of 6 of our treated dense collagen animals has metastases. So the mechanism is not the same between dense and not dense. We also find, when we go back to this gene expression array, that one of our biggest hits is this thing called prostaglandin endoperoxide synthase 2, or COX-2. And what this gene does is it converts arachidonic acid, it's part of the eicosanoid pathway. It converts it into prostaglandins and something called prostaglandin E2, involved in inflammation and cancer. When you take aspirin or ibuprofen, you're taking a non-specific drug that blocks both isoforms. There's a selective one called Celecoxib that blocks COX-2, which is the inducible isoform. So what we did is we enrolled our mice on a bit of a clinical trial with COX-2. This is an FDA-approved drug for arthritis. And what we were able to find is that while the wild-type animals weren't that much different, The dense collagen animals have increased tumor growth. These are the bigger tumors, and they regress when we treat them with this anti-inflammatory Celecoxib. The dense collagen animals have more metastasis, and that is reverted as well. So to summarize, in all the things I've told you, we have this dense breast tissue, but it's due to, in part, what's going on is increased collagen, and it's really not the amount of collagen or the amount of density, it's the structure, which then contributes to metastasis and inflammation. And so to bring this back to what we've been looking at, this will be very brief, if we think about our rope idea here, and this row rock as being one of the things that's driving all this cancer signaling, if you're relaxed, and your fascia might be relaxed as well, but you know, most of us don't live lives like that. And so I would propose that perhaps oftentimes we're all tense and we're stressed, and so our baseline level of these signaling pathways are elevated. Remember, this is part of this pathway, but I'm proposing that all of this leading to these pro-cancer genes could potentially be elevated. Now, I warn you, I'm not encumbered by any actual data on this fact. So I'm hypothesizing. So if now you go and you do acupuncture or you do yoga, you're actually stretching this out. Is that bad? That's something we have to think about. We're activating this further. But what I think might happen is now we're bringing it back to relaxation, and that the end state is more relaxed than the stress state, and that then you get back down to a protective state. So my lab does not do whitewater rafting. I'm going to have to join Boris's lab. Here we are eating sushi, all you can eat sushi. The work I told you about was Carla Espona is doing the Celecoxib work. Matt did all the in vivo, the outcomes data. Joe did the intravital imaging. Maria did the neutrophil work. And Suzanne has done the lung metastasis work, and I didn't have a chance to talk about the others. Thank you very much, and I will take questions.